That's great. Okay. Yes, as Liz introduced, I'm with ThermoFisher Scientific, and specifically, I support the visualization software in the life sciences. We have a product called Amira 3D software. There is also a technical workshop happening in the afternoon. So yesterday we already delivered one. There's also one this afternoon and one tomorrow as well. So before I get into the Amira side of things, I want to show you a quick video that at first may seem irrelevant or strange. So I'd like to just quickly do a poll of the audience. Does anyone have a clue of what this is? Does anyone know what this is? A rocket. I like that answer, but no, much smaller. It is a pen. It's a special kind of pen. Yeah, it's a stylus. Getting closer, closer. So this is an Apple Pencil, and it was CT scanned. And the reason I'm showing this animation, there's two reasons. One, to show you how segmentation looks like. So we're looking at the raw image data now with the kind of hazy orange color. But you saw there was also a surface overlaid, and that's a kind of segmentation, which is just a fancy term image processing people use for isolating regions of interest. And you do that for visualization, but also for measurement purposes. I also show this to illustrate how, as a company that services multiple industries, we have powerful visualization technology that can be used for the life sciences. So that was from our material sciences focused product called Aviso. Now, to cater to the life sciences industry, we know as biologists or structural biologists, whichever, that our data are rarely so clean. You know, that's just a fact of life. So on the left, this animation is probably not playing so well, but on the right, you're going to see these are both volumetric electron microscope image data sets that I've worked with. On the left is a focused ion beam SEM image of heart cell tissue. And on the right, we have bone tissue. So these are osteocytes that live within what are called lacunae. It's a pretty large field of view. I have the other animation pulled up in VLC. Let me see if I can drag that over here so you can actually see this. I tried to get it to work, but it wouldn't. So you can just see a cross section going right through the samples. So, you know, just quickly, even if you don't have any expertise in image processing, you can see that there's the same intensity values all throughout the image that correspond with different feature types. So kind of the simplest methods of segmentation don't truly apply here. But using the tools that I'll illustrate today, we can actually make some sense of these complex and really dense biological EM data sets. So on the left here, I'm showing how we pulled out tens of millions of individual myosin filaments. Each line here, it may be tough to see, but each of these blue-green lines is an individual segmented filament, and there's quite a lot of them. The color corresponds with their orientation, if I remember correctly, or it could be their link. I actually can't remember. I'd have to check the analysis. And then on the right, I'll show a little animation here. Let's play, please. You can see how we've isolated different regions according to what an expert observer found them to be. So we've got blood vessels, osteoblasts, and again, this was a study of the osteocytes and the lacunae that they live in. So this is, of course, a cryo-electron tomography workshop. I started with some other imaging modalities. So what does this look like when we apply Amira and the power of Amira to this data type? Now, probably many of you have seen this animation, but just to give you a taste. So this is a small section of an organelle from a single-celled algae in the genus Chlamydomonas. And so you can see ribosomes in the gray, as well as a cross-section through the Golgi body, colorized according to their function, which also corresponds spatially to where in the Golgi it's from. So Amira is used to animate and visualize all of these different segmented regions of interest. And overall, it's just a very powerful visualization method to help someone that's not immersed in the world of cryo-electron tomography to see and understand the features in your data set. Because ultimately that's our goal, right? We don't just want Cryo-ET people to look at our data, we want everybody to see them and understand them. Now, how do we actually go from the raw tomograms to that, in my opinion, beautiful looking animation? Well, it's a process, right? And Amira is not just for the visualization, but it's also for the segmentation. Now, typically, there's sort of an intermediate step you'll go through, which is broadly called image processing. So Amira is a very, very capable toolbox for image processing. What I'm about to show is another tomogram data set. These are from Harvard Medical School. They were looking at some bacteria that have a process of cell death, so apoptosis, that's very similar to what we find in eukaryotes. So that's their interest here. So what you actually are going to see is what's called a liposome with many pores inside of it. And the pores are what they were interested in. So I'm showing here as an intermediate in this animation here. Maybe I should go back and pause. This is an overlay with a membrane enhancement tool that's called Tomosegmentation. And so this is implemented in Amira software, and compared to the raw image, it's a much easier output to segment directly, either by hand or with automated tools. So you see here in the red and yellow, we're getting a really strong signal of where membranes are found. And that's what I used to actually produce an overlay here in green and blue of those pores and then the liposome edge. I also showed the edge of the carbon grid just for interest, and some gold beads. Those are not biologically interesting, so I pull those out. And this is all done within Amira software. So that's what Amira does, more or less, especially with Cryo-ET data. Following this, I have a few examples of other things you could possibly be doing with the tool. These are not all Cryo-ET data sets. So for example, here, this is a focused ion beam SEM data set, not done at cryo temperatures. It's just a fixed sample. And in this case, we have a very large volume. It's probably 10 or 20 gigabytes of data. And it's very, very heterogeneous, as you'd expect of biological tissue. So the traditional methods of thresholding or isolating the phases according to their intensity range, that wouldn't really capture much of anything here. And Cryo-ET data is very, very similar. So where the field is moving right now and where Amira is going is to lean heavily on the concept of machine learning. So machine learning is a very large catch-all term. It applies to many, many things. In Amira right now, we're thinking about pixel segmentation, which is a kind of image-to-image transformation. So the input is grayscale data like this, and the output is also an image, which in the case of Amira is a segmentation image data set. So at the upper left, you'll notice there's a little red box. That is intended to show that in the process of machine learning, typically you'll have some kind of ground truth. And this is an expert annotation of what you're anticipating from the output. And this is used to train what's called a model. So once the model is trained, then you run the deep learning with this trained model on the full data set to get some prediction of an output. The red box shows you visually how much data were used to train a model that are then used to predict a result on the rest of the image. So it's only about 1 to 2% of this data set was ground truthed by hand. Specifically in Amira, we implement what's called UNet, which is a very popular image segmentation method. We have a 2D and a 3D implementation. In this case, I use a 2D implementation. And in order to provide ground truth that are from throughout the sample, I placed landmarks that are shown with those dots, and there's squares you'll notice expanding out from them. These are the actual locations of data that I pooled together to produce ground truth information. This is really relevant for the biology domain because our samples are not homogeneous, right? If it was really homogeneous, you could say, okay, just take that little corner up at the upper left and train it on that. But in our case, like for example, you can see that there's I think this is a blood vessel going through the sample. That's not evenly distributed throughout the sample. So in order to create good ground truthing, maybe I need to over-represent in that area. So Amira gives you the power to, initially randomize all of these points, but you can also adjust them if you need to over-represent some portion of your data in the ground truth input. On the right, this is a cartoon that's often shown anytime UNet is described. This is a kind of deep learning model. It's also called a convolutional neural network. Convolution is a kind of resampling, and each blue box here is representing a convolutional layer, as it's called. The number of layers determines whether or not it's considered deep learning versus, correlation is machine learning, right? Just standard regression is machine learning, but it doesn't have a lot of layers. It's not a very complex model. Deep learning has many, many more layers, usually upwards of 10 to the more sophisticated ones that are in the news right now have hundreds of layers right now. So UNet is so called because it has a characteristic U network when it's abstracted this way. So we have an input image on the left, and then it goes through several convolutional layers. And then there's how to call it, feedback mechanism where we're targeting a certain metric. And you can think of it loosely of an accuracy metric. It's a bit more sophisticated than that. But we're trying to improve at each iteration with a lot of random guesswork along the way. What are good parameters for all of these individual mathematical operators, which are called layers? So quite a bit of computation that goes into this guesswork, but the output is pretty impressive. I remember I showed you that only 1 to 2% of that image was labeled by hand. We're now able to predict the whole data set. So this is 4 phases. In green, we have membranes of the axons. In red, I think those are the mitochondria. In yellow, we have vesicles. And then in purple, it may be hard to see it, but there's a few purple regions. Those are the synaptic clefts themselves. So the spaces between axonal dendrites. On the right, just to illustrate another way that you can use segmentation, because we now have voxels assigned to particular materials, we can view them one at a time. We don't have to view it all as one very crowded mess. We can see in 3D how only one of the phases looks like. So at the upper right, these are the mitochondria. And if you weren't aware, mitochondria are not usually spherical. They tend to actually have a pretty oblong shape. And there's a lot of variation. So you can actually see that with the segmentation data intuitively. You can also measure that, of course, but seeing is believing often for many people. And at the bottom right, I'm showing, it's a bit hard to see on this screen, but you can see the vesicles. I'm sorry, that's not right. The synaptic clefts, and each color just represents how they've been isolated independently from one another. So the colors are totally arbitrary, but this allows us, through what's called labeling, to collect measurements on each of them. So these researchers were interested in the size and the surface area of those synaptic clefts. So I mentioned that Amira is moving in this direction of machine learning. One of the newest things that we've implemented, I will show in about a 3 or 4 minute video here and talk over it. If you're attending the workshop, I'm going to actually illustrate this with you. Previously what I showed was, I guess you could call it processing offline, where I generate some ground truth, train the model, and this is not done with an expert in the loop. What is new is that we've implemented an expert in the loop kind of deep learning segmentation method. What that means is it's very difficult, and I get this question a lot. In fact, I got this yesterday, like how much ground truth do I need for a model? And I like to always say, like, every model is wrong. Some models are useful, right? So how to know whether your model is useful often requires a bit of iteration. You may start with a certain amount of ground truth, train a model, see what that looks like. And as an expert observer, you'll know, oh, I noticed that these areas are false positives or false negatives. So I should include more examples inside my training data set. So this is an actual snapshot of the Amira interface, specifically our manual segmentation workroom. For returning users, we're in the process of updating this workroom, so it might look a little bit different, but the logic is mostly the same. And we were placing landmarks, what are called patches. Those are those squares. That's the input information used to train a deep learning model. So the person recording this video is painting microtubules with a very, very steady hand, apparently. And you see that they're only labeling a super tiny fraction of this data set. It has about 80 slices in it. And I think in this animation, they only really label just some portions of this one slice and then create a deep learning model based on that. So it speeds up in a minute here. You don't have to watch this person go through every single step. But yeah, they're just moving up and down through the slices to show you the quantity of data. All right, so they do more and more. And then eventually they're going to click on a button that basically allows for a very, what we call shallow network machine learning approach, which is just a same as the UNet, but there's fewer layers, which makes the whole process much faster to train. And this allows you to do sort of an expert in the loop kind of behavior. So we experimented with this yesterday. I think it was pretty efficient. So if you're in the workshop today or tomorrow, you'll see the same thing. You'll be able to experiment with the same data set. Okay. So they're just showing off how you can go through all the patches and see them in 3D. All right, now we're actually going to train the model. So there's a one button, you click for that and click, create a selection. This is sped up just so the video finishes in a reasonable period of time. But in our experience yesterday, I think on my hardware, it only took about 2 minutes. And in red, you see a highlight of the prediction. And that's on all the slices. So with not a lot of effort, we're producing already a reasonable first guess. And you can disagree with that. That's the whole point. If you don't like these results, you can update them. We give you all the tools to edit the selection directly in this interface, or your approach might be to produce more ground truth. Now, this is a material by material thing. So if you have multiple materials like vesicles or mitochondrial membranes, you could go through those material by material. And this is an active area of development. So if you have feedback for how this could work or should work, definitely let me or one of our thermo colleagues know about that. I think we'd be interested in talking to you. So that's what's specific, I would say, to cryo-electron tomography. Kind of zooming out a little bit, Thermo has a full range of tools. As you know, we're using the hardware and the software that comes with the microscope itself. Amira is an offline tool. So you can do this data processing at any point. If you have the right software access, you could do it from home if you wanted to. It's taking your raw image data. In the case of Cryo-ET, that would be your tomograms. So they've already been aligned and reconstructed in iMod or some other software. And through image processing and segmentation, you're going to pull out regions of interest for presentation. And it shows meshing down here as someone else's slide. I don't know if you'll actually ever do that, but definitely the measurement and presentation of data is all done within Amira. Now, the application itself is not specific to cryo-electron tomography, which I think is a strength for people that are doing any correlative work, or if you're ever in the future doing imaging work that's not cryo-electron tomography. So it's worth knowing that we can do molecular visualization in the software. If you have models from the protein data bank, for instance, you can view those inside of Amira. There's a molecular visualization technology built into the software. We can also do what I call large volume analysis. That's the focused ion beam SEM, or if you use an ultramicrotome. This is on a timer. That's what happens when you borrow slides from other people. So yeah, this is large volume analysis, much larger compared to the TEM that you're working with. And then if you're working with CT data, which is how I got my start in visualization, Amira is quite capable of working with those data types as well. We're also not focused on a particular sample size. As I mentioned, you can do molecular visualization. This audience would be interested in the angstrom to nanometer scale, but we're quite capable of also working with organ or even much larger sample analysis. As I mentioned, if you have other modalities, we can do correlative work inside of the software, following up excellently, I think, on Jay's talk from just a moment ago. We can do 3D image registration or 2D image registration, although most people get Amira for its 3D capabilities, of course. And we're working well with MRI data, diffusion tractography, synchrotron CT, you name it. Just another flavor slide of the kinds of things we're doing with Amira. And then specifically for this audience, as I mentioned, in the workshop, you'll get an experience with that same data set I showed a moment ago. So you can see how we're using AI to do the segmentation of these cryo-electron tomography data. And overall, I really enjoy using the software, talking about it, teaching people. I'm going to be here today, the whole day, and tomorrow as well. So if you have visualization questions, you can tap me on the shoulder at any time. I'm also joined by my colleague who's in the middle in the light blue shirt, Ken, or you could reach any one of the Thermo people. We'll point you in the right direction. So with that, I think we have quite a bit of time for questions. Thank you very much.
Great, thank you, Alex. Do we have questions to start off? Okay.
A couple of questions.
I have a question about so when you do the segmentation, especially the deep learning one, and then generally you will segment something like is exactly what you're looking at, right? And then the microtubule is, I would say, easy because you can actually trace.
Mm-hmm.
Have you ever tried that on actins, especially those very densely packed actins? And then also, like, after you get your segmentation, how would you actually verify it's segment correctly, especially when your eyes cannot help you?
Yes. Yeah, two good questions. So the first question was, in the case of actin, you're just saying actin filaments, right? Yeah, okay. So actin filaments are much smaller than microtubules. So at the resolution of cryo-electron tomography, they're not going to show up as cleanly. So in that case, because they're more or less tube-like, or filament-like, there's a technology that I showed for the focused ion beam data of the muscle tissue. I'm kind of struggling with my mouse here. Hold on a second. Well, I'm having a hard time navigating. I don't want to go through back on the slides, but we have a image correlation based method for tracing tube-like structures, which I would recommend instead of deep learning, because I think it's a better fit for that problem. And you can mix and match these things, so you don't have to do one or the other. So that works by translating a template, which is a tube, just a very abstract cylinder, to FFT space, and your image is also placed in FFT space, and there's a cross correlation that's matched between your data and the template using a sliding window, and this produces a correlation image and an orientation image. You pair those together and you can trace filaments. The advantage of that is because you expect not to find a very high correlation when you don't have pixels that are correlated in like a tube-like shape. They're like beads on a string or something like that. So the method is very efficient in my experience, as long as you parameterize it correctly, and that requires practice, just like anything else. But some of the slides I showed right at the end, like this, these red traced areas are not from voxel segmentation, like the deep learning, but it's actually using this tube tracing algorithm. So I think it's the better approach for that problem. The second question was more, how do you verify that your segmentation is good or not? And that's a chicken and egg problem. So in that case, what one can do is generate some kind of artificial ground truth and test the algorithms to see how good they are. To my knowledge, that's really the only approach that the image processing community finds acceptable, I guess you could say. That's like the gold standard. But oftentimes with Cryo-ET, we're delving into the unknown. No one's ever looked at this sample before in this way. So it's a hard problem to solve. I'm not really sure how to say it other than the only way you can be 100% sure is with some kind of simulation of data and perfect ground truth to compare against. But usually that's not going to resemble your sample. That's at least with the tools I know of that are out there right now, really the only way you could know for sure. If you have something else in mind, I'm happy to speak with you offline about it. I think, though, that's the best approach. Two more questions, at least.
Hi, thanks. Very interesting. I'm just curious, kind of following up from the previous question, does Amira come with kind of preset models for a microtubule, mitochondria, Golgi, whatever that should be in mammalian cells, pretty standard? Is that something that you have?
Yeah, that's a good question.
Yeah, you're thinking about?
Yeah, it's a good question. That's the vision for sure. I think deep learning models, though, they train on features that may feel very obscure to human eyes. What I mean by that is the contrast and the intensity range from microscope to microscope, or even imaging session to imaging session, may be different enough that the model doesn't behave super well on your data. So I would say it's sort of somewhere in between an academic target and industrial, like commercial target, to build up a repository of ground truth segmentation to develop robust models that can apply in many user data cases. Right now, in my experience, it's more efficient to train your own model in this way, and that's why I show off that tool. I have tried to use other people's pre-trained models on data that weren't like their demonstration data, and I don't know. Sometimes I'm not very satisfied with the results. So I like to have the ability to train my own model. There is a problem of overfitting that is always there when you talk about extrapolation of a model onto unseen results. Again, any statistics class teaches you that about correlation. You know, if you extrapolate way out here, are we still making the same inference, or should it be more bounded? Deep learning is that, but a lot more complex, but also not really. So the models that you generate from one data set are probably going to be overtrained on that specific data set. So the ideal model would be trained using a very large number of data sets with similar features in them, but different imaging conditions, maybe different microenvironments. I'm not sure what the right term is, but I like where your mind's at, because I would like that to exist too, to greatly accelerate this whole process. We're not quite there yet, but this is also kind of a call to action. If you want to help commercial software producers, if you upload data to Protein Data Bank or the electron microscopy database, and you have segmentation data, please also include the segmentation data. And if it's okay with the people you work with, make sure that the usage rights are available for commercial use, because oftentimes, like after I filter like the millions of data sets that are out there to available for commercial use, it's like 10, you know, there's like nothing. So the choice was made by whoever uploaded the data. It's not really my choice, but I'm not gonna go against someone else's will there. But that would be useful for us in the commercial software space to have more data that were commercially available, just as a general comment.
Hi, I was just curious, when you're actually doing the ground truth and you're setting the stage for that, manually picking features, is there a strategy one uses in which features you are in fact highlighting in that ground truth? You start with the larger or smaller structures, the more difficult
you can certainly spin your tires segmenting the whole image, but that may not be that useful to you. So your project will probably focus on one or two features. So certainly focus on those. Maybe you want a membrane for context of something. You know, the data I shared a moment ago, they had vesicles, they had mitochondria, lipid membranes, and some microtubules inside of them. So the final result, it shows you all of those things because you can see them more or less with your eye. We are very interested, I would say, as a company in going in the direction of this particle placement inside of the tomograms. We're not quite there yet. I think people in this audience are more knowledgeable of that space than I am. But it's really quite catered to your goal. So I wouldn't approach it as a let's segment the entire image. That's a lot of work, and you may not really benefit from that in terms of your project goal, unless your point is to create, you know, like a beautiful, fully segmented data set. And you know, we do those here and there, but in practice, you're usually segmenting one or two features, and that's it. To answer like a spacing question, or how much is there a hole in this membrane, something like that. That's a question where you don't have to segment the entire image to answer that question. So it's usually more of a time consideration. How much time do I have, or how many students do I have to do this work with me or for me? That ends up being your limiting resource rather than the technical ability of the software to do it. Did I answer your question? Okay. Yes.
Hey, so maybe this is the meshing tool you briefly mentioned, but is there like an easy tool for converting, like, say, a membrane or some of your segmentation data to like a oversampled array of points that you could import as like seeding subtomogram like coordinates?
Sure. Yeah. Amira has a lot, and I really do mean a lot of features. And one thing you could do is find, let's say, for any individual object you see segmented here, I could pull out a point that represents its center of gravity, and you could export that as a point cloud, which is very, very versatile. You could use that. You could also do that on membranes or tube-like structures. So you could have like a sheet of points. Is that what you're thinking about?
That answers my question.
So the answer is yes. I would say there's not really a tutorial for something that's specialized, like pre-built. But as a commercial software, you ask us questions and we'll get back to you within a business day. That's what we try to do at least. So we won't leave you hanging. Just ask us and we'll tell you if it's a quote unquote big ask, in which case, we may not have the final answer within a business day, but we'll at least get you some kind of response quickly. Okay.
Cool.
How much more time do we have? I have no sense of that. It's 11:38. Okay, just keep going, I guess.
Alex, I had some questions from online and I was going to combine
Great.
two of them together. One of them is a question about if you had a different scale. So if you were at the level where you're seeing a tissue image and you could see different cells such as neurons and glia,
Right.
could you use the segmentation tools in Amira to differentiate at the cell level, not just the organelle level?
Absolutely.
Okay.
Yeah.
A follow-up question is, what is the smallest scale? Is there a limitation for things such as seeing different shapes of molecules, such as ribosomes? Would you be able to do segmentations that could identify different conformations?
Yeah, in terms of the software, there's not a firm limit on the physical size of your data. In software, we talk about, how much data does it take in memory? That's a more relevant limitation. When it comes to protein level segmentation, though, often you're going to reach for what's called particle picking, and that currently isn't implemented in Amira. It's been talked about for years and years and years, but our sentiment is a lot of other programs exist to do it, and they do it very well. And we've just decided not to make it a commercial target. But if enough people ask for it, then we could do anything. That's how it is in a commercial group. We can't do everything, but we can do anything. So we just need to have enough people asking for that to implement it. And thus far, we just haven't had enough people, especially from the commercial user base. I know this is mostly an academic group, but commercial users just don't seem to care that much about that. As relevant as I know it is, and I think it's a cool method. Yeah, so for instance, in this illustration, the ribosomes, which are illustrated in Amira as surfaces, they were found and rotated according to an external software, actually, full disclosure. And the illustration is done in Amira, but their coordinate space was found using a particle picking software of some kind. Yeah. So if you take a note of whoever asked that question, if they want to follow up with me or someone in Thermo, we can see if that's something we should be incorporating into the software more directly.
So in terms of measurements that you mentioned, right?
Yes.
Could you identify, let's say, the top left corner, this two nuclear pore complexes, right? Can you, after you segment the whole volume, can you let Amira tell you where are the, let's say the nearest or the furthest points on that pixel? And how is that achieved?
Yes. Yes, I was pretty light on the measurements discussion in my talk, but that's what a lot of people use Amira for. It just is, it ends up being very specific to each person's use case. So the kind of spatial in 3D measurement, that's definitely possible in Amira. Right now, those nuclear pore complexes are interpreted by Amira as molecular models that were converted into surfaces. So the surface is a discrete point at which we can cast a distance field out of. And then we sample whatever other point, how far away was it from the nearest surface of the nuclear pore complex, if that makes sense. So you can represent that as a number, or you can visually see it as well. So you can actually colorize like how far away points are. Since it is a visual tool, that can sometimes be more interesting or useful to you. So if you have a specific need, please ask me. I've helped people do that exact thing before. So I know it's possible. Again, it's one of those things that's a little too niche to have a direct tutorial on how to do it. That just ends up being how it is when you have kind of a general purpose tool. It's hard to write a guide for every single person, but please ask me, and I'm sure we can do a lot of the things that I've been asked about. Any other questions?
I
No?
guess not. Well, let's
Okay.
thank Alex again.